the Forschungszentrum Jülich.
There you go.
And yeah, I'm going to talk today about phages and the relations between phages and aminoglycosides. So should I start or should we wait for Jessica?
No, let's, if you're ready, then let's start.
Okay, then.
Okay, I will stop my share and I'll let you take over.
Yes. Okay. All right. So yeah, first of all, thanks a lot to Jessica, Jan, and Stephanie for organizing this PHAVES seminar series and for making this possible. And I'm very happy to talk today.
Oh, and if anyone has any questions, please enter into chat and we'll be able to get to them at the end of the presentation.
Okay. So I think most of you already know that if you suffer from a viral infection, such as the flu or cold, you should not take antibiotics because antibiotics are said to be ineffective against viruses. And today I want to challenge this idea with you and show you that in some cases, antibiotics are actually able to inhibit viral infections. So much like humans, bacteria have their own viruses, which are called phages. So I guess most of you, if not all of you, are already familiar with phages. So I won't go into great details to introduce phages. But I would just say, and I think we can all agree on this, that phages represent a permanent and ubiquitous threat to bacterial communities in nature. And on the other hand, we have bacteria which have an extensive and diversified chemical repertoire. You can see on the right here the genome of a Streptomyces strain, and on the right-hand side, the different biosynthetic gene clusters which are encoded in its genome. And you can see that in this example, this genome can encode 11 different biosynthetic gene clusters. And these numbers can be even higher with some species of Streptomyces with up to 20 or even 25 of such clusters. And still, the true function of these clusters is often not known. So putting these 2 points together, it's quite tempting to ask whether some of these metabolites could be used by bacteria as so-called antiphage metabolites. So I already mentioned briefly Streptomyces, and since I assume that not all of you are familiar with this bacterium, I'm going to introduce it shortly. So Streptomyces are gram-positive and aerobic bacteria that represent the largest genus of actinobacteria with more than 500 species, and they thrive in soil and decaying vegetation. And Streptomyces have been studied for two main reasons. The first one is that they have a very complex secondary metabolism. And in some cases, like as shown in this picture, you can see the pigments, the different metabolites with the naked eye, since these metabolites are colored and visible, yeah, as I said, with the naked eye. And the second reason for which Streptomyces was studied a lot is because of its complex developmental cycle. So most bacteria, they divide by binary fission, but this is not the case for Streptomyces. It had instead this cycle where it starts as a spore, which first germinates and then develops into a network of interconnected cells, which is called the vegetative mycelium, which then serves as the basis for the erection of an aerial mycelium, which then differentiates into chains of unique genomic spores, which are then released in the environment and can start a new cycle. And importantly, this secondary metabolism and the development cycle are strongly connected with each other, because most of these metabolites are secreted at specific periods of this cycle. Okay, and so back in 2018, so essentially at the start of my PhD, we wanted to isolate new Streptomyces phages. So I went into the forest around our research center and isolated phages from soil samples. And in total, we described 5 new phages. So 2 of them infecting Streptomyces venezuelae, and 3 of them infecting Streptomyces silicular. And these 2 species are being, let's say, one of the model species of Streptomyces. And just as a side note, for those of you who are Star Wars fans, the names should probably sound familiar. And something which was quite striking and which we noticed after isolating these phages was that for 3 of these phages, so for these 3 Streptomyces silicular phages, we noticed that the plaques were colored with, the plaques were surrounded by colored halos, which suggested that, Streptomyces cerevisiae was reacting to phage infection by releasing these pigmented compounds, and that these compounds could potentially be involved in defense against phages. And right around that time, so basically a few weeks afterwards, came out a very interesting paper from the lab of Karen Maxwell, in which they showed that anthracyclines, which are a class of antibiotics produced by Streptomyces, are potent inhibitors of phage infection. And so this led us to wonder whether this could be only the tip of the iceberg, and potentially many more antiphage compounds were waiting to be discovered. And a few months later, we observed by chance that phage infection was inhibited when we added to the cultures apramycin, which is an antibiotic belonging to the class of aminoglycosides. So what are aminoglycosides? They are bactericidal antibiotics, which are active both against gram-positive as well as gram-negative bacteria. And so the different members of this class have sometimes slightly different mechanism of action, but overall they all inhibit bacterial synthesis of proteins, and they do so by binding to the 16S ribosomal RNA. Here you can see on this model on the right, so you can see the ribosome, and if we zoom into the A site of the small subunit of the ribosome, you can see in red paranomycin, which is an aminoglycoside binding to the 16S ribosomal RNA to the 16S ribosomal RNA, and this leads to incorporation of incorrect amino acids in a nascent protein. And in terms of history, the first aminoglycoside to be isolated was streptomycin, and this was a major breakthrough in the history of medicine, since it was the first antibiotic active against tuberculosis. And in nature, aminoglycosides are produced by different Streptomyces species. And back in the 1940s, 50s, and 60s, there were a few reports about the inhibition of phage infection by streptomycin. And so for all these reasons, we were interested in aminoglycosides and wanted to study their potential antiphage properties. So in nature, usually the producers of natural products are resistant to the produce that they release. And this property is very important when we want to study potential antiphage molecules, since any strong effect on bacterial growth would prevent the appreciation of an antiviral effect. So this is why the first thing we did was to construct aminoglycoside resistance strains carrying plasmids encoding resistance genes. Then we cultivated these strains and then challenged them with phages in presence of different aminoglycosides. Here are the results, some of the results we obtained with Streptomyces venezuelae and the phage alderan. And you can see in absence of antibiotics, a strong plaque formation. So yeah, this plaque formation reflects the lysis of the cells by the phages. And you can see that if you add to these plates agromycin or apramycin, you can't see plaques anymore, meaning that phage infection was completely inhibited by these two antibiotics. And similarly, we did the same assays with E. coli and the model phage lambda. And yeah, in a similar way, you can see that addition of kanamycin or apramycin strongly reduces the plaque formation of this phage. And here on the right, you can see the overall results of the screening, where essentially the darker the color, the stronger the inhibition of the phages. And you can see on top of the caption that we used a diverse set of bacterial hosts, so both gram-positive as well as gram-negative, as well as diverse phages belonging to the main phage families. And overall, we observed at the same time the strongest and also the most consistent inhibition with the aminoglycoside apramycin, and with two phages in particular, so the phages alderan and the phage lambda, which are the ones I already showed you on the left. And so for these reasons, in the next slides, I'm going to focus on apramycin and these two phages. So all in all, the screening on plates showed that aminoglycosides were able to inhibit phages infecting widely divergent hosts, both gram-positive as well as gram-negative. So the screening I showed you before was done on plates, and then we wanted to move to liquid cultures to see if the inhibition we saw could be also observed in liquid cultures. So here's an example of infection performed in microtiter plates. And you can see with the blue curve that when you add the phages alderan to these cultures of Streptomyces venezuelae, there's a strong growth defect of the cultures, which reflects the successful amplification of these phages. But then if you add apramycin to these cultures, you can see that with the red curve, that this lysis is not present anymore. And actually the curves, so the red curve, which is the infected cultures, is overlapping almost perfectly with the uninfected cultures, the gray ones, meaning that infection was completely inhibited by the addition of apramycin. Then we also wanted to perform infection in the macrophagic system, in which we inoculated spores of Streptomyces and infected them with phages. And you can see on the left infection in absence of any antibiotic. You can see that the mycelium is completely lysed by the phages. And in the right, you can see that when apramycin is added to the culture, barely any lysis is visible. So yeah, all in all, these results show that apramycin is able to completely inhibit infection with the Streptomyces phage alderan in liquid cultures, as well as on plates. And so far, the experiments I showed you were performed with purified antibiotics that were bought at commercial suppliers. But then we wanted to see whether we could observe this inhibition, this time with spent medium of natural Aminoglycoside producers. And so for this, we cultivated Streptomyces tenebrarius, which is the natural producer of apramycin, and harvested spent medium, so basically splenation of the cultures at different time points. So the first thing we did was to verify the amount of apramycin by ACMS. So with the red curve, you can see that after 1 day of cultivation, there was no apramycin detected in the cultures. But in contrast to that, after 2 days, as shown with this blue curve, there were significant amounts of apramycin detected. So once we confirmed that these cultures contain apramycin, we wanted to test the effect on phage infection. So we performed again infection in macrotiter plates. And you can see here, there's a similar graph to the one I showed you before, where you can see that addition of phages causes a strong growth defect of the cultures, which reflects the successful infection in these cultures. Then when we add to this fresh medium, spent medium harvested after 1 day, you can see that so the cultures are a bit shifted, the curves are a bit shifted, but overall there's still a stronger phage amplification. And however, when the spent medium we added to the cultures was harvested after 2 days, so as shown with this blue curve, you can see that the uninfected and infected curves are once again almost completely perfectly overlapping, meaning that phage infection was inhibited by addition of the spent medium. And to verify these results, we wanted to determine the final phage status of these cultures. And so for this, we spotted the splenation of these cultures on the bacteria alone and determined looked at plaque formation. And you can see that when the phage infection was performed only in fresh medium or in fresh medium supplemented with spent medium harvested after 1 day, there was strong plaque formation, meaning that phage amplification was successful. And in contrast to that, when spent medium harvested after 2 days was added to the cultures, you could not see any plaque anymore, which confirms that this spent medium was able to completely inhibit phage infection. So yeah, so altogether, these results show that spent medium of an apramycin producer was able to reproduce the effect of purified apramycin. And at this point, you probably ask yourself, what's the mechanism of action? And in other words, which type of the phage life cycle is inhibited by apramycin? And so here is one diagram showing the main steps of the phage life cycle, lytic phage life cycle. And so to infect its host, a phage must first adsorb to the bacterium and then inject in its DNA, which first becomes circular and then is being replicated, transcribed and translated to form new viral particles, which are released upon cell lysis. And so what we did was to look basically at these different steps to see which one was inhibited. So first of all, we performed an assay called a potassium reflex assay. And so in this assay, the entry of the phage DNA correlates with transient efflux of potassium from the cells to the surrounding medium. And so this is how this assay is basically a proxy for the injection of phage DNA. And these are the results we obtained. So in black, you have infection of E. coli with lambda. And the black arrow represents the addition of the phages. And you can see that after some time, the specific potassium voltage, which is basically, which correlates to the amount of potassium ions in the medium, is increasing, which means that from the cells were escaping from the cells to the medium, so which shows that the infection was successful. And in blue, you have the negative control with delta Lumby mutants, and Lumby being the receptor of this phage. And then we did the same, but this time with apomycin. And you can see that overall, the curves are very similar, meaning that, the injection of phage DNA was not impaired by the addition of apomycin. So then we moved one step further and then looked at the replication. And so for this, we measured the amount of intracellular phage DNA over time by qPCR. And you can see with the blue bars that the amount of intracellular phage DNA is increasing exponentially over time. And whereas when apomycin was added to the cultures, shown with the red bars, the amount of intracellular phage DNA is actually increasing much earlier and more to a linear rate. We then had a look at transcription. And for this, we performed RNA sequencing during phage infection and then mapped the reads to the phage genome. And you can see that in absence of apomycin, there's a strong buildup of reads at both 90 and 180 minutes after infection. And if you do the same this time in presence of apomycin, there was basically barely any reads detectable. So these experiments indicate that apomycin is able to prevent both replication and transcription of the phage genome. And then we wanted to confirm these results by some microscopy. And so we performed fluorescence microscopy, in this case with E. coli and the phage lambda. And so here, blue represents the bacterial DNA, which is stained by DAPI. And the red color corresponds to the phage DNA. And after 90 minutes, there are like this red foci appearing, which correspond to phage DNA being replicated. And there's also this strong increase between 30 and 90 minutes of this phage DNA. And if you perform infection, this time in presence of apomycin, you can still see this very faint red foci at 30 minutes, but they don't increase in intensity at 90 minutes. So you can see here on the graph that the intensity of this red foci is not increasing, which means that phage DNA was indeed integrated inside the cells, but this phage DNA was not amplified. So yes, at this point, we have a good idea of which step of the phage life cycle is inhibited by aminoglycosides. But because aminoglycosides are well-known inhibitors of bacterial translation, there could still be the possibility that the effect we see is not due to direct effect on the phages, but rather because of an indirect effect where just because these antibiotics block the translation of the bacteria. And so to completely rule out this hypothesis, we performed the experiments I'm going to show you now. So one measure way by which bacteria can be resistant to aminoglycosides is by modifying the aminoglycosides. And this is the same for many other antibiotics. And this is, by the way, the way we used to build the resistance strains I mentioned in the beginning. And so how this is working is that these enzymes, they add chemical groups to their corresponding aminoglycosides. This can be either acid group or nucleotide groups, as well as a phosphorus group. And this modification leads to a strongly decreased affinity of these molecules to the 16S ribosomal RNA. And so as a result, these molecules have basically lost their antibacterial properties. And so we wondered what is the influence of these modified aminoglycosides on phage infection. And for this, we took again our beloved apomycin and modified it with one enzyme called AAC345, which catalyzes the acetylation of the 3 amino group of the 2 deoxysubtamine ring of apomycin, and which leads to so-called acetylated apomycin. And so we performed this reaction in vitro with pre-fat proteins, and we checked the efficiency of the reaction by ACMS. And once we had performed this confirmation, we wanted to test the effect of acetylated apomycin on phage infection. And so something important that I want to mention here is that in this case, we used the wild type. So Streptomyces phonetically wild type. Although in all of the experiments I showed you before, we always used resistant strain. And we use this wild type because normally the inactivated apomycin should not have any effect anymore on the growth of the wild type. And here, so in this panel, we added apomycin, so the regular apomycin, which is non-acetylated. And you can see with these gray and red curves that there is essentially no growth at all, which is normal because the wild type is highly sensitive to apomycin. Then we performed again the same infection, but this time in presence of acetylated apomycin. And so as expected, you can see that with this orange curve, that the growth is essentially not impaired anymore. Because as I said, the acetylation causes the loss of the antibacterial properties of apomycin. But what was more interesting and quite striking was that, as shown with this so yellow-green curve, that here there was basically no lysis anymore caused by phages, meaning that this acetylated apomycin was still able to completely inhibit phage infection. And so, yeah, as I said, this experiment showed that acetylated apomycin still strongly inhibits phage infection, although it has completely lost its antibacterial properties. And so with this, I'm already at the end. So I hope I could show you that aminoglycosides are able to inhibit phage infection in a widely divergent hosts, both gram-positive as well as gram-negative. And so, yeah, we can say, so I think it's quite interesting to see that one molecule can have seemingly completely unrelated effects, which are in this case antibacterial and antiphage effects. And yeah, then we could show that this inhibition could be reproduced using span medium of natural aminoglycoside producers. And this is quite important because it could suggest that the inhibition we observed could be also relevant in a natural setting, so in nature essentially. Furthermore, we also, so concerning the mechanism, we could show that the blockage caused by aminoglycosides occurs early on in the phage life cycle, namely between injection and replication of the phage DNA. And lastly, we showed that the acetylation of apomycin suppressed its antibacterial but not antiphage properties. So yeah, to come back to the cartoon I showed you in my second slide, I think we can say now that certain antibiotics, such as aminoglycosides, can treat viral infections in some cases, such as viral phage infection. Okay, so now just a bit about future perspectives. So yeah, so far there were 3 main classes of antiphage molecules described recently. So anthracyclines, as I already mentioned in the introduction. Then we also have viperins, which are chain terminators, meaning that they produce modified nucleotides, which are then triggering which are interfering with the transcription of the phage mRNAs. And this was shown by the lab of Oberheim. By Oberheim and colleagues. And yeah, lastly, there were aminoglycosides. And I think this list is to be continued in the future. Yeah, just as a side note, so I was looking into reviews about the roles of natural products in nature. And it was quite shocking to me, at least, that in many cases, phages were not even mentioned, which I found very surprising, given that, that phages are super crucial to bacterial communities. And yeah, so I'm believing that the field of antiphage molecules is probably going to boom in the incoming years. And for this, I think we would need, it would benefit a lot if people from different fields moved in, because so far, most of the labs, so essentially all 3 labs which made these discoveries are primarily phage labs. And I think it would be nice if, for example, people from the natural products field are moving in, and because they could bring new skills. And yeah, as the last point, because of course, phage therapy is always important when we discuss about phages. Yeah, the inhibition of phages by aminoglycosides we described could be also relevant for phage therapy, because if you imagine that you want to combine in like this combination treatment phages and aminoglycosides, it would be nice to check first that the phages you used are not inhibited by the aminoglycosides. And so with this at the end of my talk, so this work, the work I presented you was a true team effort. And so I'm very thankful towards the many people who were involved in this study, both inside the Frunzke lab as well as outside. But I want to thank 2 people in particular. So first of all, Aël Hardy, who is my co-first author on this paper, which did a really fantastic job and actually made many of the experiments I showed you today. And last but not least, Julia Frunzke, my supervisor, for constant and enthusiastic support at every step of this project, as well as of my PhD in general. So yeah, thank you for your attention, and I'm happy to answer any of your questions and to discuss about phages in general.
Wow, thank you so much, Aël. That's really cool. Yeah, let's go straight to the questions. Okay, so Ergen Akturk asks, do you have any proof that apomycin inhibits the infection of phages, or can it be interfering with the replication mechanism of the phages? So you kind of answered that, but could you go into a little bit more detail about, like, do you know how it's inhibiting the replication mechanism?
The short answer is no, we don't know. So far, we only have an idea of which step is inhibited. Okay. So if I develop a bit more, I think, so our idea is that, so aminoglycosides are, let's say, versatile molecules which are able to bind to a wide variety of nucleic acids. So it's only been described, like, for example, they can bind to ribozymes, to like a diverse set of nucleic acids, both DNA and RNA. And so for me, it would make sense that the effect we see is due to the binding of this aminoglycosides to the phage DNA, and this at an early step in the life cycle. So yeah, as I said, so after injection but before replication. And yeah, there's actually a potential parallel between aminoglycosides and anthracyclines. So because this was shown by the lab of Can Maxwell that that in this case anthracycline also seem to inhibit the same step of the phage life cycle. And actually it would make sense because the phage DNA at this stage is quite sensitive to any molecules, because it's not protected by any proteins usually, and it's also not so it's linear and also not compacted. It's a relaxed state. So I think, to me at least, it makes sense that this incoming phage DNA is the prime target for aminoglycosides, but we have to show it. We haven't shown it yet.
That's really cool. Okay, so Reagan asks, what could have caused a silent increase of the apomycin levels during phage replication while studying the mode of action?
I'm not sure I got the question right. Can you repeat the question, please?
They're saying, what could have caused the silent increase of apomycin levels during phage replication?
I mean, so in the slides I showed you, we didn't. So there was no increase in phage in apomycin production during phage infection. So I think maybe the person was referring to the slides. Oops. So in the slides I showed you, it was here basically. So maybe I got the question wrong, but what we did first was to grow this streptomycin producing strain. Sorry, not apomycin producing strain. So we let it grow, and we harvested the spinetomes at different time points. So in this case, 1 or 2 days. And then we used this span medium and then added this to the cultures and then measured the effect on phage infection. So it was 2 different experiments. And so I can also say that so far we don't have phages which directly infect this apomycin producing strain, so Streptomyces univalus. We tried to get some phages, but I have to say it's quite hard, and we're not sure whether because it's just because apomycin just inhibits all the phages, or because for some other reason this strain is poorly infected by phages. But yeah, this would be very cool to have. And so far we don't have such phages infecting this apomycin producing strain.
Interesting. So have you been able to like remove the antiviral properties at all of apomycin, or like is that?
So in the slides I showed you, we tried one modification. So we tried this one acetylation. Yeah, here. So there are many different enzymes in activating these aminoglycosides, and so in the example I showed you, this acetylation was abolishing the antibacterial properties, but was not having any impact on the antiviral properties. But this is maybe not the case with other enzymes. So this we are testing now. We are increasing our set of enzymes, and we are actually very eager to see if this thing we observed was just kind of an accident, or if it's the case for all of these enzymes.
That's cool. That's really cool. Lonely Susie asks, would it be possible that bacteria such as the actinobacteria or actinomyces produce novel compounds in response to phage infection or predation?
Yeah, this is, I mean, so I would say my gut feeling is that there are many more waiting to be discovered. So we are also working on different molecules when we haven't really found anything convincing until now. But I think for sure I would say there are probably many more compounds, so antiphage molecules which are there. And also I think my feeling is that so maybe we will also rediscover some already known molecules, meaning that the molecules that we will find will probably not be novel, but they will be non-molecules, and then we didn't know their true function in nature, probably because at least my feeling there are many molecules so which are known, but usually the real function in nature is not known. And I think to me it would make sense that at least some of these molecules have antiphage properties.
Got it. That's really cool. That this is like a huge new world. And then so David Ngugi asked, do other antiphage systems come into play even in the background of AG, or actually do the bacteria encode other antiviral mechanisms?
It's a very good question. So we didn't have a look at this specifically, to be honest. So we had a look into the antiviral arsenal of Streptomyces venezuelae, and I think, if I'm not mistaken, there's at least one restriction modification system, and probably I forgot, but probably one CRISPR-Cas system, but I'm not sure if it's functional. So I mean, the short answer is that I don't know, but yeah, I think it's very interesting now. I think now a lot of focus in the phage community is to study not the systems as related, but to look at them together. And yeah, this I mean, it would be really interesting to see how these small molecules interact with other like well-known defense systems, such as restriction modification systems or CRISPR-Cas or other ones.
Yeah. Wow, so it's got a huge web of interplaying effects. And Michael Skurnik, sorry, asks, do you see a dose dependence on the phage inhibition?
Yes, we do. So for apomycin, I mean, I can go back to the screening here.
Yeah.
So you can see that for the ones which show an effect, so essentially, mostly, for example, agromycin, gazogamycin, apomycin, and to a lesser extent also kanamycin. I don't know if it's super clear in this graph, but there's definitely a dose-dependent effect. And for example, for apomycin, so this is on plates, but in liquid cultures, we can already see an inhibition at 1 microgram per ml, and then this inhibition becomes complete at 10 microgram per ml. And so yeah, I think we can safely say that there's definitely a dose-dependent effect.
Wow. Well, okay, so we have one last question from Newton. Newton, do you want to ask in person, or do you want to type your question in chat? Oh yeah, I have to give you permission, sorry.
Yes, greeting to the presenter and the organizer. Yes, I would like to ask the presenter if he has managed to culture alone or to see the effect of aminoglycoside on the phages alone.
The phages alone.
Alone.
On alone or alone?
No, alone. It's just
alone. Okay.
Not in culture.
So at some point we wanted to look at, we wanted to do some, I think, cryo-EM to look at if, for example, the aminoglycosides could like cause structural changes in the phages, in the phage capsids. I think we didn't do it in the end. And to me, I think it's maybe not the most likely explanation. But we didn't have a look at this yet, at least.
Yes, because you just allow me to make this contribution. You know that phages replicates more when the bacteria is at an exponential phases. And you know that the aminoglycoside inhibit, maybe they interact with the translation process of the bacterial host.
Yes.
So it means they have the antibiotic in this case are eating in layman language. In layman language, they are eating the food of the phages. So if they are eating the food of the phages could not really replicate in this condition. So there is a competition between them. Therefore, I may recommend that you just add the antibiotic to the phages only, not in culture with the bacterial hosts. And then you also have a control whereby the phages are in SM buffer, you know, the buffer which is appropriate for the storage of phages. So perhaps from that one, we may really see or find out whether the antibiotic has the antibiotic did they do inhibit the growth of the phages. That should be my contribution.
Yeah, thanks a lot. I think to answer your point, I think on paper it's possible to, as you said, to incubate before infection just the phage with the antibiotic and then filter out the antibiotic. The problem is that, if you do this, I mean, if this interaction between the phage and the antibiotic is reversible, then you will, in most cases, I would say just when you do this, when you try to incubate the phages and then wash out, you will get rid of the antibiotic. And if you don't wash out, you will still have the antibiotics when you mix the phages and the bacteria together. So I think it's quite tricky to address, but something else I can say is that with these experiments I showed you here, so here we used a wild type. So, and something important, maybe I didn't stress it enough, is that here the apomycin we used was acetylated. And so here it has completely lost its antibacterial effect. It cannot bind to the ribosome anymore. So by this, I think it's a quite good proof that the effect we see is probably not due to the direct inhibition of translation, because this acetylated version of apomycin is not able to inhibit translation anymore. I hope I answered your question.
Okay, and then so there's a couple more questions. I guess a little bit more time before we have to close out to breakouts. But Denise Romero asks, do you know something about apomycin toxicity?
So I mean, let's say, so if you use a wild type strain, so I mean, so of course it doesn't impact all bacteria, but I would say for apomycin, many gram-positive as well as gram-negative. So in our case, both E. coli and Streptomyces venezuelae, and actually the other Streptomyces species, most of them are inhibited by very low amounts of apomycin. So I would say even if you use the wild types, lower than 1 microgram per ml probably is enough already to inhibit growth. And so exact mechanism of action, I don't know if this is what the person wanted to know, but I would say, as I already mentioned, the primary cause is the interference with translation. But then I would say that then the following steps is a bit debated. So people used to say, I think in the 80s and 90s, that the toxicity was mostly based on the incorrect proteins which were synthesized, because if you interfere with translation, you can get then essentially proteins with wrong sequence, and this could have then toxic effect downstream. I think now it's a bit moving from this. I'm not entirely sure what's the exact current state of this question, but I would say now it's more based around the potential. I saw papers about that this inhibition of translation was then interfering with the potential of the cells compared to the medium, and this would have then further effects. So I forgot the exact details of the papers, but yeah, I hope this is answering the question.
And Miklos Gernick asks again, can you isolate AG-resistant phage mutants? Or have you done anything to that?
Yeah, we tried. I would say it's not trivial. So because I think it's a good idea because we, at least this is how people usually do when they have new defense systems. They try to isolate mutants. So phage is able to overcome these defense systems, sequence them, and then see where the mutation lies, and then have maybe potential ideas of how this system is working. So we tried this by forming infection in small amounts of apomycin and then increasing slowly, and then see if there were phages still so able to infect in higher amounts of apomycin. I think so. This was not really conclusive, and one potential explanation is that if we assume that that apomycin and aminoglycoside in general, that they bind to the phage DNA and this way they interfere with replication, and then it's possible that you cannot change this from the phage by just one or few mutations. It's let's say it's an intrinsic property of phages, and so it would not be easy for the phage to just mutate one of its proteins to overcome this inhibition. So it's maybe one of the reasons why we didn't manage it so far. And of course, there could be many more. But yeah, it's definitely a good idea. And we also tried, so far we tried mostly on liquid cultures, but I think it's also interesting to test some place to see if maybe this works this way.
Oh, so, okay, that's really interesting. Wonder, okay, so I'll just ask these other people's questions. Jivan says, this might be a silly question, but did you check the identity of the bacteria before and after treatment or combining apomycin and the phage?
Okay, the identity of the bacteria. I'm
not sure what that means. I guess did you sequence it before and after, or?
No, we didn't. I mean, I don't expect the bacteria to suddenly change during infection. I mean, we didn't. No, I mean, we didn't do this. I think, yeah, there's no clear reason why the bacteria should change during phage infection. I mean, of course, you can have mutations, but this should not strongly influence. I mean, yeah, maybe I didn't understand correctly the question.
Yeah, I guess these. So I have a random question. I guess we got to close out soon, and Jessica will take over and do the breakouts. But I have a question. Is it so the apomycin producing bacteria? Were you able to like give that? Sorry, as a non-biologist, I'm just really curious about this. But like, were you able to give that those capabilities to some other bacteria that doesn't produce apomycin and see whether that would prevent phage infection?
Yes, it's a really good question. Yeah, I mean the biosynthetic cluster of apomycin is known. So it's like 30 kb essentially, yeah, more or less, with all the genes necessary to synthesize this molecule. And so yeah, we actually considered this to take this whole cluster and put it into Streptomyces venezuelae, for example, and see if in this way Streptomyces venezuelae could be protected. So yeah, this is something, yeah, I think quite interesting, because in this way you can really, yeah, narrow it down to one molecule. So because, for example, in this experiment I showed you with the spinatins, there were many other compounds. I mean, of course, So, I mean, we are pretty sure that it's apomycin causing this effect, because in the slides I showed you before, we had purified apomycin, and we saw the inhibition with just the purified apomycin. So I think it's what the effect we see here is most likely due to apomycin. But yeah, there are of course many more, for example, molecules in this spinachins, and the best way to get rid of them would be to just express this cluster on its own in the different bacteria.
That's so cool. All right, Jessica, any thoughts?
Thank you so much, Aël. I'm glad I got to catch the second half. You guys, nobody needed me here, so I'm glad it all went out on a pitch.
I'm almost a real biologist now.
Yeah, he's an honorary microbiologist. Can I share my screen just quickly before we go to
Sure.
breakouts? I have a slideshow. Okay, do you see my screen?
Yep.
Yep.
Yeah. Okay, yeah, just wanted to let everybody know that our next PHAVES is coming up with Dr. Saima Aslam on the 20th of September in a couple of weeks. So this is switching over to the medically focused version of PHAVES, which we're in the process of rebranding, which we may call Phage MD, but this is still under the auspices of PHAVES. So come and listen to Saima give a talk about her experience treating patients with phage. And then just a quick shout out to our sponsor for this webinar, Phage Futures Europe. This is happening. Many of you have been in the past. Aël, you attended this with us one year as our social media person. So it's happening in person again. Yeah, and it's in Brussels, Belgium, 23rd to 24th November. So you can register for that. And we're going to be doing some promotion of that, and it's always a great conference. So definitely check that out if you're interested in translation of phage research. That's all I'll say. Okay, so I'm going to create the breakout rooms, but anyone who's heading out now, feel free. If you don't feel like showing your face, you can leave. And maybe someone will be lucky enough to get stuck with Aël in a room and you can ask more questions. But yeah, so I'm gonna just have a look at the number of people that are staying so I can create the right number of rooms. So we're gonna do 6, let's do 5 rooms. So you can just click to join the room when you get a little notice. And then we're gonna switch you out after 5 minutes and then we'll re-randomize you in with some other people.